Thank you so much for inviting me, and thank you all for coming. I had a lot of fun preparing this talk. This is a topic that I get to spend maybe 10 minutes or so on when I teach Bio 301 genetics. And now I have a whole hour to dive into the depths of craziness. So I'm excited to share with you what I learned. And if you didn't pick up one of these handouts as you came in, I encourage you to do that. One side has an outline, so you know in general where I'm going, and the other side has typical sexual development for humans. And that would be really handy as we go along, figuring out how you can have manipulations of various aspects of these pathways and what happens. I want to start with a woman named Maria Jose Martinez Patino. She was a Spanish hurdler. In 1986, she qualified for the Olympics. She qualified in a trial. And she also took a chromosome test at that time. And she was declared ineligible for the Olympics in 1988 because she was not XX. She's XY. She has something called complete androgen insensitivity syndrome. So complete AIS is what this is. It's about 1 per 20,000 live female births that we see this. And usually it's diagnosed at puberty. So a young child is born, looks female, has typical female characteristics, and then at puberty does not menstruate. And they're brought in, and at that point in time learn that they are not actually XX. There's a mutation in complete AIS in a gene that codes for the androgen receptor protein. The androgen receptor protein is the protein inside your cells that responds to testosterone. Testosterone is the hormone that is really turning males into males. So here you have a human being who's making testosterone and is XY, but the body can't tell. The receptor for testosterone, that androgen receptor, isn't functioning. So it doesn't recognize or respond to testosterone. What you see with this genotype then is a completely normal exterior female, but there are internal testes, perfectly normal testes in the abdomen, not descended. They do have breasts, they have a short vagina, they don't have any uterus. And adult women that have complete AIS are usually a little taller than the typical woman. They're not as tall as the average male, but they are taller than the average female. So how does this interesting combination come about? And here's where I'm going to pull up your pathway. So you can follow along on the back of that handout while I talk to you a little bit about it. We'll start with the first 5 weeks of fetal development. A fetus is born, begins by having bipotential gonads in those first 5 weeks. And in the absence of particular chemical signaling to make them become testes, the gonads develop into ovaries. This pathway is kind of considered a default pathway for the gonads to become ovaries, but there's been some question lately about whether that's really as default as we think. I think for the purposes of this talk, we'll consider the female development pathway to be default. It happens in the absence of male signaling. So for a gonad that has bipotential to develop into a testis, there's this whole cascade of events that has to happen. And it's all initiated by a single gene, SRY. SRY stands for sex-determining region of the Y. It is a gene on the Y chromosome, just as it sounds. And what SRY does, it turns on at about week 6 or so. This gene produces a protein that then goes and turns on lots of other genes. One of the other genes that it turns on is SOX9, and I'm showing you that in your diagram. SOX9 is not on the Y chromosome. It's on a different chromosome. I have to look this up. It's on 17, chromosome 17, SOX9. SOX9 then initiates an entire cascade of other events, and eventually that bipotential gonad develops into a testis. Once there is a testis, once the cells in that gonad are testis cells, they secrete testosterone. Testosterone causes a different bipotential tissue to become a penis instead of a clitoris. And testosterone also is needed to cause the descent of the testis out of the abdomen and into the scrotum. So if there's no SRY gene, then that bipotential gonad develops into an ovary. And interestingly, the female ovary doesn't have any documented role in the further differentiation of genital tract structures in the female. Not all of the structures in the fetus are bipotential. I've talked about a couple that are. But interestingly, at first, the fetus actually has rudimentary structures for both a uterus and a vagina, and seminal vesicles in the vas deferens. So some of these need to further develop, and some of them need to regress for the typical development of human sex. The fetal testis is responsible for secreting various hormones that stimulate the growth of male duct structures, and it secretes some other hormones, not testosterone, that cause degrading of the female uterus. Meanwhile, if you've got an ovary, the uterus, the vagina, the fallopian tubes, they develop even if you don't have to have, even if there's no other signaling present. And then at puberty, we have the testis secretes even more testosterone. So you have growth of the penis, the shoulders broaden, the muscles grow, hair in more places. And if you have ovaries, the ovaries secrete lots of estrogen, and so your breasts develop, the hips broaden, and hair in more places, right? All right, so that is what we're going to start with from typical development. And I'm going to walk you through a few more ways that you can create trouble with these pathways. If you're looking back at the other side of the sheet, you'll see at the bottom I have some notes about different ways we can talk about sex. There's chromosomal sex, the X, the Y. You can also talk about gonadal sex, whether you have a testis or an ovary. Physical sex, what do you look like? What does your body look like on the outside? And then there's really sort of brain chemistry sex. How do you perceive yourself? Do you perceive yourself as being male or female? So usually these all agree, and it gets interesting when they don't. So with complete AIS, what I talked about at the very beginning, we have agreement with the chromosomal sex, XY, and the gonadal sex, testes, although they're internal. And then you switch. The physical sex, the exterior, looks female, and the brain chemistry, most complete AIS individuals see themselves as female. So a disagreement between these four types. One more thing before I leave AIS. AIS is not always complete. Sometimes there's something called partial AIS, and this results from different types of mutations in the androgen receptor protein. So if you have a mutation that completely makes it non-functional, that's complete AIS. If there's a mutation in that gene that just makes it not so good at doing its job, then you can have partial AIS. And there's a broad range in that. It can be as simple as a typical male that has low fertility. So there's a lot of variability. Taking you back to Maria Patino. She's actually the first athlete to formally protest the chromosome test. And the IAAF agreed. They saw the right that there was no advantage for her body because even though she had the Y chromosome, her body was not using the testosterone. So they switched. They no longer test for a Y chromosome. They changed instead to testing for high levels of testosterone. And they only excluded athletes that had high levels of testosterone and were able to respond to that. So, Maria Jose Martinez did not qualify for the next round of Olympics in 1992, alas. She missed the hurdling by 0.10 of a second. She's now a professor of science education in sport at the University of Vigo in Spain. I'm going to switch now to another way to be an XY female. AIS was 1 per 20,000. This way is called Swyer syndrome. It's not as common, it's 1 per 80,000 live-born females. Same as AIS though, it's usually not diagnosed until puberty when these individuals do not menstruate. And there's two different ways this can happen. And I think you should flip back to your side that has the pathway on it because this will help. So remember, this is an XY that's going to become a female. The two ways that Swyer syndrome can happen. One way, a mutation that inactivates the SRY gene. So the Y chromosome is present, but the SRY gene on it doesn't work. Another way, a mutation that inactivates all the copies of SOX9. SOX9, remember, that's not on a sex chromosome, it's not on X, it's not on Y, it's on chromosome 17. But if you inactivate SOX9, you also do not have enough of the male cascade pathway to develop into a male, you develop into a female, externally at least. So Swyer syndrome, we have the XY genotype, you have a non-functional development pathway for the gonads, so the default pathway, the female pathway, happens. You get a functional uterus, quite interestingly, and a vagina, but no ovaries. And I know I said that you should get ovaries. This goes back to this question of is that pathway all that default? It's unclear why you don't get ovaries, but what you get is something called a gonadal streak, an undeveloped clump of tissue. And it doesn't produce any hormones. So there's not any puberty. They look female, but then at puberty, they don't menstruate and they don't develop the secondary sex characteristics. So typically, women with Swyer syndrome are treated with hormone replacement at puberty. And then we see normal development of breasts and other secondary sex characteristics for females. And what's really cool is they can become pregnant with a donated egg. Their uterus and vagina are functional and fine. And there's at least 15 reports in the literature of successful pregnancies for individuals with Swyer syndrome. Oh, one more thing about Swyer syndrome, they are also tall. Back to the being not as tall as the average male, but taller than the average female. So both Swyer syndrome and complete AIS have that. One more thing about XY women before I leave this topic entirely. When you look at elite athletes, let's take a look at the Olympics. In 1992, there were 7 XY women per every 1,000 women in that Olympics. In 1996, there were 3 XY women per every 1,000 women in the 1996 Olympics. In the general population, there is 1 XY woman per 20,000. So there's a really high prevalence of XY among elite female athletes. And this is really curious. We really don't know why. There's some suggestion that it has to do with height. Perhaps the fact that the Y chromosome does have other genes besides SRY, so some of those genes affect height, and certainly some sports, height matters, but not all sports. And we don't necessarily know what all the genes on the Y chromosome are doing, so there's still more to learn about why this is happening in elite female athletes. All right, time to switch. So let's talk about the other way that you can have sex reversal if you are XX but male. And I've talked a lot, so it's time for you to do some educated guessing. I'm going to give you a little time and think about this, looking at your sheet over here. Come up with at least one way and talk to your neighbor. How might this happen? How could you be XX but male? I think the people who took genetics have an unfair advantage. All right. I heard both of the right answers, or 2 answers that are correct while I was out there. 2 that I'll talk to you about. The first one that I heard was, what if you have excess SOX9? So you are XX, but you have a lot of the SOX9 protein for some reason. Yes, that is one way you could be an XX male. And usually what happens is it's not so much you have more copies of the SOX9 gene, it's usually you just make more protein from it. It's being overproduced from that gene. This is kind of curious because overproduction of SOX9 apparently doesn't affect XY individuals. If you are XY and have extra SOX9, you're completely normal and fertile. And in fact, there are cases in the literature where someone who is XX inherited from their dad an overactive SOX9 gene. So if you are XX and you have lots of SOX9, then yes, you have trouble with infertility. All right, so this XX male with extra SOX 9, usually not diagnosed until adulthood. So externally, they look very typically male. They are always infertile. They don't have any sperm at all in their semen. And why? Why is that? Probably because there's no SRY. So they have no Y chromosome, so there's no SRY gene. And the SRY gene turns on a lot of genes. It doesn't turn on, I know I just put SOX 9 on the handout. It doesn't turn on only SOX 9, it turns on a lot of genes that are important for male development. So, oh, and one more thing, there are some other male-specific genes on the Y chromosome, not just SRY. And of course, those are missing. There's no Y chromosome. So we end up with infertility. Also, the testes are often not fully descended and somewhat small. These individuals are a little bit smaller in height than typical males. Their height and weight is more typical of an average female. Okay, the second way. And if you're keeping track, you'll notice that I did not write these on your handout. So if you're looking, you see that I left you some space. All right, the second way. The translocation of the SRY gene off of the Y chromosome and onto the X chromosome. So a translocation is just an exchange of genetic material between two chromosomes. In meiosis, we get something called crossovers, where chromosomes trade bits, and it's a regular thing, and it's supposed to happen, and it's great. However, meiosis should not trade bits between the X and the Y chromosome, or at least not this bit. So on occasion, there's a crossover that swaps the end of the X and the end of the Y chromosomes, and you end up with a Y chromosome that doesn't have any SRY, and an X chromosome that has the SRY gene on it. This is actually the most common way to be an XX male. 90% of XX males are what we call SRY positive, meaning they have SRY even though they're XX chromosome. Again, usually not diagnosed until adulthood. So externally, typically male. However, they are always infertile. Again, no sperm in their semen. And why? So now we have SRY. Well, back to this idea of there's no Y chromosome, and we know that there are some male-specific genes in the Y chromosome. Those are probably needed for proper sperm development. Again, they're a little smaller than typical males. They look height and weight more like females. And for those of you who took genetics, I found this a really interesting thing. You know that in individuals with two X chromosomes, one of them is always inactivated in a bar body, right? And it should be random. This is not happening in XX males with the SRY on the X chromosome. There's preferential inactivation of the chromosome that doesn't have SRY. So we have this non-random X chromosome inactivation. The X that carries SRY is not the one that's made into a bar body. So, sidelight for those of you who are really into this stuff. All right, what about extra sex chromosomes? That's the next topic on your list. We have, what if you have 3 sex chromosomes? An X and an X and a Y? Or what if you have an X and a Y and a Y? So this is often a topic that people have heard a little something about, and I'm going to let you talk to your neighbors about what have you heard about either XXY or XYY. All right, so the one I'm hearing from all of you is that I've heard of Klinefelters. I think I might know something about that, or might have heard about it before, so we'll start with that one. That's the XXY. This is actually the most frequent chromosome disorder in males. It happens in 1 out of every 650 live male births. However, most men remain undiagnosed through life. We think that more than 60%, more than 60% of men with Klinefelters don't ever know they have it. And if it is diagnosed, it's usually after puberty and for reasons of reduced fertility. Not infertility, but reduced fertility. And there really aren't any distinct features for it. The testes are usually somewhat smaller than average. Testosterone level is on the low normal range, And so sometimes, if it is diagnosed, they're treated with a little extra testosterone, and this does help further development of the secondary sex characteristics, but it doesn't change anything about their fertility. And moving on to the other one, XYY. And I didn't hear very much about this one out there, so it might be something you have heard less about. It's sometimes called Jacob's syndrome, sometimes just called XYY syndrome, and it has another name, and I'll get to that in a minute. This one is estimated to be 1 in every 1,000 live male births. And again, in most men that have it, the extra copy of the Y chromosome causes no problems whatsoever. More than 95% of guys who are XYY, I think, don't realize that they have it. So it's often not diagnosed. And if there is something diagnosed, it's often misdiagnosed as something else. These individuals, they do have accelerated growth when they're young, and they're quite tall as adults. The average height is 6'3". However, they have normal sexual development and occasionally reduced fertility, although not always. More recently, we've figured out that these individuals have a higher risk of developing or having learning disabilities, and they have a higher risk of ADHD, the attention deficit and hyperactivity disorder. Okay, so now the other name for XYY is supermale. Anybody heard about supermales? Maybe. Okay, we don't talk about them that way anymore because, alas, this is kind of a myth. So XYY was first reported in the literature in 1962, and when it was reported, it was thought that maybe this extra copy of the Y chromosome would make men more violent and more aggressive, and they called them supermale. And so in the 60s and 70s, shortly after it had been reported, scientists went looking for men that were XYY in places like mental institutions, maximum security hospitals, prisons, and they did find a higher percentage of XYY men in those places than in the general population. But these studies were not well designed. They were not good. The sample sizes were too small. Things were not well done. So later on, people did larger studies, better studies, and what they could confirm for us is that XYY men, they do have more convictions for crimes than XY men, but not more convictions for violent crimes. And recently, there's been some suggestion that maybe what's going on is that XYY men have a higher conviction rate because they were not helped with the ADHD and the other behavioral disorders that come perhaps with some of the XYY genotype. So there's something about XYY for me. I think. Yes, I'm going to skip the next one. I had Turner syndrome, which is just the X chromosome. I'd be glad to return to any questions if somebody wants to hear more about it later. But I'm going to skip that. Turner syndrome is a female that has only one X chromosome and nothing else. We're going to jump over to hermaphrodites. So what about hermaphrodites? A true hermaphrodite. None of the things I've talked to you about so far fall in the category of hermaphrodite. I know it sounds like they should. They're partly male, partly female, but scientifically, a true hermaphrodite is an individual whose gonad has both ovarian and testicular tissue. So this is about 1 in 20,000 live births. The testosterone levels are usually sort of intermediate between male and female ranges, and the external physical genitalia are usually quite ambiguous. And back to this mixture of ovary and testis, there's kind of two ways. Some people who are hermaphrodite have an ovary on one side and a testis on the other. And many more of them have something called an ovotestis, which is a single gonad with cells from both types mixed together. 60%, or maybe a little more, of true hermaphrodites are XX, and they typically lack the SRY gene. So it suggests that maybe there's something else going on. Maybe there's overactivation of a gene that's normally turned on by XRY, or SRY in these people. But we really don't know. I can say, though, that about a third of hermaphrodites are mosaic. Some cells have XX, and some cells have XY. And it appears that this was from a fusion of two fertilized eggs very, very early in the pregnancy. So, the ovotestis, usually the testicular part of the ovotestis is not functional. But the ovarian tissue part is sometimes functional enough to produce oocytes. And typically they do have a uterus, most of the time. There are no reports in the literature that I can find of any cases where both types of the gonadal tissue are fully functional. So there's no self-fertilization that we know of. What about treatment? When a child is born as a true hermaphrodite, there's always a question of how to respond about this. Typically, there's been a partial gonadectomy. So they remove in early childhood either the testis part or the gonad, that's the ovary part, so that at puberty, only one of those two things is directing development. Early on, it tended to be that male gender was assigned and the ovary part was removed because externally these individuals have something that looks more penis-like than not. But now, actually, there's been a trend for assignment of female gender because of the fact that the ovotestis usually has viable eggs and a functional uterus. So there's a chance for that individual then to live life as a fairly functional female, to have eggs and bear children. Recently, there's been some discussion about a third way. What about delayed gender assignment? Is it possible to simply leave well enough alone for a while? But there's no data, or at least it's scarce enough that I can't find it, about hermaphrodite children and what's happening there, or how hermaphrodite children perceive themselves and how this relates to their further development. All right. So we've talked about chromosomal sex, X's and Y's. We've talked about gonadal sex, ovaries and testes. We've talked about physical sex, what you look like on the outside. We have not yet talked about brain chemistry sex. How do you perceive yourself? So what do we know about the genetics of being gay or being lesbian? My short answer to you is not a lot. Sorry. I can say to you that there's a lot of studies, multiple studies, that have confirmed that homosexuality, both lesbian and gay, is absolutely partly genetic. There is a genetic component. That is confirmed. There are several studies of identical twins and fraternal twins. So let's start with sisters, for example, comparing identical twin sisters with fraternal twin sisters. So identical twins are exactly alike. They share 100% of their DNA. Fraternal twins share only 50% of their DNA. But twins, on whichever you are, have the same environment. Okay. So identical twin sisters are much more likely to both be lesbians than fraternal twin sisters are. So if one of the sisters is a lesbian, it's much more likely for the other one to also be lesbian if they're identical than if they're fraternal. And the same goes for brothers and being gay. So there's clearly something genetic. All the environmental factors being equal, genes do matter. But not all pairs of identical twins are both gay or both lesbian. So it's not just genetics that determines sexual orientation. There has to be an environmental role as well. I'm going to actually leave lesbians for a while. We know almost nothing about lesbian genetics. I can tell you a teensy-weensy bit more on the genetics of gay men, however. There's a handful of small studies. It's a really complex area of research. Of course, it's fraught with lots of social concerns. But I'll tell you what I do know, what I can find. So 2 to 5% of all men are gay. In 1993, the very first study published using a very small sample size, and they reported that they found a region of the X chromosome that was linked to being gay. No specific gene, just a particular region, and that was in 1993. In 1995, another study using a different sample, also small, confirmed that finding. Then in 1999, another study, different sample, again, small sample, did not confirm the finding. They didn't see that. So there's conflicting evidence about whether or not there is or is not some little region on the X chromosome that contains a gene that affects whether or not someone is gay. In 2005, there was another study, again, another small sample. They found a region on chromosome 8 that was linked. Again, no specific gene, just some region that contained a gene that must have some influence. And then in 2014, finally a big study, a big sample, and yay, they found those two regions, the same two. They found the same region on chromosome 8, and they found the same region on the X chromosome that had been found in 93 and 95. So we're feeling better about there's something on chromosome 8, and there's something on the X chromosome. What? I don't know. But I can speculate for you. So these are really big regions. There are lots of genes in the regions that have been implicated. And so this is pure speculation. It's not my speculation, it's pure speculation of the people who wrote the papers. It's really interesting speculation though. So let me start with chromosome 8. In the region that was linked to being gay, there is a gene called neuropeptide BW receptor 1. It's expressed in the brain, and in mice, it has been shown to have a big effect on social interactions. So maybe different versions of this gene could affect social interactions in human, which then might lead to social interactions that tend to be gay. On the X chromosome, there's a gene called cyclic nucleotide gated channel alpha 2. It's expressed in the brain, and it's been shown in mice to affect odor-evoked sexual behaviors. So we know that odor, sense of smell, has a role in sexual behaviors in many animals, and probably in us too, although we don't really think about that very much. And so again, maybe a version of this gene, an allele of this gene, can result in a slightly different perception of odor and a slightly different response to that odor in sexual behaviors in humans. One more cool gene, also in the region on the X chromosome, is something called MAGE11. And this produces a protein that's a co-activator of the androgen receptor, the receptor that responds to testosterone. So maybe an allele of this gene, a version of this gene, either enhances or decreases the androgen receptor's response to testosterone, and therefore affects a person being gay or not gay. Again, I want to highlight here that there is no actual evidence yet that any of those three genes are actually important. And also want to remind you that genetics is, yes, involved, but not the only thing. There's certainly environmental factors as well. I'm going to leave genetics for just a moment and take a little detour into physiology, because there's something that I found really interesting in my research. It's called the older brother effect. In males, homosexuality in males has been consistently and repeatedly linked to the presence of older brothers born to the same mother. So what this suggests is that it might be some sort of progressive immune response on the part of the mother. So some sort of antigen produced only by a male fetus, and as the mother has more and more children, if they are male, then her response to that, her antibodies build up over time. And the increasingly accumulation of these antibodies would interfere more and more with the fetal development of subsequent sons. It's kind of like the Rh factor thing, for those of you who think about that. There's pretty good evidence that the particular protein might be neuroligin 4. I wouldn't say it's absolutely secure, but neuroligin 4, the gene for this is only on the X chromosome, so only males make it. It's a male-specific protein. Neuroligin 4, as the name might suggest, it's found in neurons of men. So it presumably has an effect on brain chemistry. There's a study done just really recently in 2017, and after statistically controlling for total number of pregnancies, they found that mothers who have gay sons have much higher antibodies against neuroligin-4 than control women. And in fact, there's sort of there is a progressive increase in the amount of antibody against neuroligin across groups of mothers. So mothers who have no sons don't have antibodies against neuroligin. Mothers who have heterosexual sons have some. Mothers who have one gay son and no other brothers, so only one son and it's gay, have a little more. And mothers who have more than one son and the youngest of them, or a younger one, is gay, have the most antibody against neuroligin-4. So I find that really interesting. However, the mother immune effect cannot explain everything about being gay. All right, a final word here, because no biology talk is complete without saying something about evolution. And so I'm going to say a final word here about homosexuality and evolution. So when you think about evolution and natural selection, natural selection typically eliminates factors that reduce reproduction. And gay and lesbian individuals don't reproduce. So why is this not being, this is a paradox, right? Why is this not being removed from our population? Well, there is some support for the hypothesis that the gene, or a gene, that influences whether someone is gay is on the X chromosome and has an advantage for women in increasing their reproduction. So the same gene with two different functions. In men, it makes them more likely to be gay, and in women, it makes them more fecund, able to reproduce better. And the other evolutionary speculation about this is kind of back to thinking about how families work. Having individuals in the family who do not have children of their own provides more care for children of brothers and sisters and other relatives in the family. So this is, if you've heard about the grandmother effect, having grandmothers around to care for children makes families better. Well, maybe having gay or lesbian individuals in the family makes the family overall better because there are more caregivers for the children. So that's a possibility too. And I have come to the end of my slides. So, I'm going to lose myself on it. So I would be glad to take your questions. I probably can't answer most of them, but I will do my very best to answer questions you guys have about this topic. Okay, you guys have to holler at me. Yeah. Okay, I think I can. That's as far as I can go. You can yell it.
What exactly is the single X chromosome?
Oh, okay, Turner's syndrome. Yeah, that's a good question. So, Turner's syndrome, these individuals, they are female. I'm going to open my slides because I could make it up, but it'd be better to tell you all the right stuff. Okay. Here we go. So, this happens 1 in every 2,000 live female births. It was described first in 1938. It's quite variable in its severity. There are two main features. Individuals with Turner syndrome, besides the fact that they only have one X chromosome, they're generally short, and they have something called ovarian failure during childhood. So their ovaries begin to degrade, atrophy, go through apoptosis, ovarian failure during childhood. So it's actually quite unclear why the ovarian failure happens, but the short stature is thought to be because of a gene on the X chromosome that has a role in skeletal development, and individuals with only one X chromosome would have only one copy of that gene, and in females, maybe having one copy is not enough. Maybe in males it's unregulated, maybe there are other genes on the Y chromosome that compensate males for height, but there's a height, a bone gene on the X chromosome. Turner's is sometimes diagnosed right after birth, sometimes not diagnosed until adulthood, all because of the variableness of it. And because of the ovarian failure, they usually require hormone replacement therapy at puberty. so that they can go on to develop breasts and the typical secondary sex characteristics of a female body. And they can have some other health issues too. They have some cardiac difficulties, bone anomalies. However, affected women can have children with a donated egg. Their pregnancies are higher risk because of the cardiac anomalies, but they can have children. Absolutely. I think that's all I have about Turners. Other questions? Yeah, shout it out.
Okay, so this is going to sound really silly, but it is actually a serious question. Are you familiar with the case of the man with two penises?
No. This question is, am I familiar with the case of the man who has two penises? I am not. So, how long ago? Was this recent?
He's currently alive right now.
He's currently alive.
He came alive with two fully functioning penises.
Huh. That's really interesting. I have not heard of that. So it sounds to me like it would be a developmental pathway problem as opposed to an X chromosome or a Y chromosome problem, but I'd have to look into it. I don't know. It's really interesting.
He did the, I don't know if you know what it is, he did all the AMA on there where he put a bunch of questions from people. That's really interesting stuff.
That's cool. All right. So he's done some of his own work figuring out what's going on. Yeah. All right. I'll have to look this up. That sounds really cool. Other questions? Yeah.
I heard that the clitoris and the penis are just different. They're differently developed. They're the same thing, developed differently.
Very good, you are right. Yeah, the question is, so is it true that the clitoris and the penis are the same tissue, just develops differently? Yes, that is quite true. Yep, and so the growth of that tissue becomes a penis, or the sort of lack of growth of it remains a clitoris. Yeah. Others? Yeah?
So there are other extra sex chromosome phenotypes. Also like triple X.
There are.
Just a little more about that.
Okay. He says, so he's heard of some other genotypes, for example, triple X. And there's also, in addition to the Klinefelters, there's triple XY as well. The more X chromosomes you have, the stranger things get. This is true. However, it never gets really crazy because X chromosomes beyond the first one are typically inactivated as a bar body. And so if you're triple X, then you have one chromosome that's active and two bar bodies in every cell. And that turns out to be fairly normal. Others? Or at least your body is fairly normal. I don't know if that's normal, but your body is fairly normal. Yeah. Yeah. He's talking about the back. It's kind of a pathway, a map.
It has a line going down from socks and pathway. It looks like it's inhibiting.
Oh, yes. He's asking about, so I put something on the pathway that I didn't talk about. He says there's this line that goes down from SOX 9, and it's got a little cross on the bottom. And he says, is that inhibiting? It is. That's quite right. So SOX 9, in addition to turning on genes and causing activation of things, also inhibits some genes involved in female development pathway. Yes, it does. Yes, it does. I think there might be a few other things on that pathway sheet that I didn't talk about, but I realized I had too much to say and I didn't want to overwhelm you. Yeah? How do you get to be XYY? Great question. When it happens in the development of sperm, when the cell that's going to develop into sperm undergoes meiosis, it should first copy all its DNA. So then it should have 2 X's and 2 Y's. And then it should divide twice. And so the first time it should put 2 of those in one cell and 2 of those in the other. And the second time, you should put 1 in each cell. So you end up with 4 sperm, 2 of them X, there's an X sperm, there's an X sperm, and a Y sperm, and a Y sperm. However, if one of those divisions doesn't go quite right, you could end up with a sperm that has 2 copies of the Y chromosome, and a sperm that has no copies of the Y chromosome. If the sperm that has 2 copies of the Y chromosome fuses with mom's egg, then you're X, Y, Y. If the sperm that has no copies of the Y chromosome fuses with mom's egg, then you have Turner syndrome. Good question. Well, thank you all for coming. I enjoyed getting to talk to you about this. I hope you thought it was